All right, so last week we talked about heredity and we talked about Mendelian genetics and traits and the things that Gregory Mendel had discovered. He used a very quantitative method, and the basic rules of Mendelian genetics does apply in general, but then we started to see that some traits didn't seem to follow that, some inheritance didn't seem to follow it. So while it's true that the chromosomes are randomly assorted in the genes, if they're on different chromosomes, all that kind of stuff, those things are true. However, the more you study genetics, the more you realize things are more complicated than just simple Mendelian genetics in the sense of how the traits respond. And there's a lot of different things that disguise the Mendelian segregation of alleles, and that is due to a variety of factors. And here are some of the ones that we're going to discuss, like continuous variation, pleiotropic effects, incomplete dominance, environmental effects, epistasis, and codominance. So these are different factors that disguise Mendelian inheritance. That doesn't mean that, of course, those chromosomes aren't randomly sorted and things like that. The first one is continuous variation. And this is the idea that more than one gene is involved in making a trait or a phenotype. In pea plants, it was kind of a little bit simpler in the sense where you can have the tall pea plants or short pea plants. And I'm sure there have been environmental factors involved in that as well. But we notice when we look at the heights of this room, for instance, or in people in general, that there's a bell-shaped curve to height. And that's because more than one gene is involved. It's multiple genes that are involved. And so this is where we can have what you call continuous variation. Not everyone is either tall or short, but there's a variety of different heights. And again, this is due to multiple genes contributing to the overall phenotypic trait. And again, the environment, it isn't just environment versus genetics. It's not just nature versus nurture. But you've got to understand that it's a combination of the two. You might have tall genes, but if you don't get a good diet, you're not going to get particularly tall necessarily. So there are multiple factors involved. But again, more than one gene is involved in making height, for instance. So that's a polygenic trait. There's also pleiotropic effects. This is when alleles have more than one phenotypic effect. So, for example, cystic fibrosis is an inherited disorder that affects chloride transport channels. And what it does is it makes the fluids around mucus areas less liquid and more thick. Now, people might say, wait a minute, he has cystic or she has cystic fibrosis, their lungs are clogged up. I may think that's the phenotypic trait, but in reality, that trait of not making the fluids more liquidy and easier to expel can happen in multiple organs. And so that, again, disguises the appearance of Mendelian inheritance. So pleiotropic effects is when the phenotype is affected by multiple organs and it just kind of disguises it. So for instance, sweat glands will look like very salty sweat glands. It won't just be the typical salty, but extra salty. Their lungs will have a lot more mucus and cause them to have more respiratory infections. So these might appear as phenotypes, but again, it's due to this chloride channel being blocked that helps to make the juices more liquidy and more fluid. The liver gets blocked and their fat digestion is affected, vitamin deficiency, night blindness. All these nasty things happen with cystic fibrosis. And again, it's a mutation affecting the ability to make the fluids and what are being expelled outside the cell more watery. There's also things like incomplete dominance that also hide Mendelian genetics. This is when we have dominant genes and recessive genes, but the reality is that the dominant trait is does not express itself fully. So in the case of like snapdragons and these flowers here, you can see that there's a red trait and a white trait flower, but there's an incomplete dominance. There isn't a red flower or a white flower in that next generation like we saw in pea plants. Instead, we see some pink plants. And so the way they describe or show the Punnett Square for that is in the following. Here we have the first generation where we've bred true for red or white flowers. And so because the trait is incomplete, we do a superscript, and there can be other ways to do it. This is just one manner. And then the next generation, they're all pink, not red or white, like you might have predicted, looking at the typical Mendelian genetics where they'd all be red, for instance, if red was the dominant trait. So in other words, the dominant trait is not as fully expressed anymore. It's not going to be a pure red. And so here's our gametes being separated out from these heterozygous individuals that are pink. And so we see the CR represents the original red gene. CW represents the white gene for the sperm, because remember meiosis, we'd have gametes. Eggs, we have gametes also. So we have superscript red and superscript W. And then we do our Punnett Square, and we see that one is red, two are pink, and one is white. These are the homozygous traits. And again, the heterozygous traits show this incomplete dominance where the red color or the white color is not, is backed off. So it's not as strong of a trait anymore because of the presence of the other gene, which can be a little confusing when you think about codominance, which is another example of something kind of like incomplete dominance, but in which case both are expressed, and I'll show that in a moment. Also realize that a lot of traits can be influenced by the environment. Like I told you about height, food, and nutrition can affect it. Well, in the case of coat colors in this snow or Arctic fox, when there's a snow environment that's cold, the gene will show the hair as being more white. When the environment is warmed up, and this has to do with daylight links and all sorts of things as well. Like when we lose trees, all those kind of environmental factors influence the genes. But the Arctic fox fur becomes pigmented in warm weather. And this is a great Darwinian kind of selection thing, right? I mean, this is a much better camouflage when there's snow on the ground, and then when the snow is melted, this is a much better camouflage. So again, evolution still acts upon these Mendelian ideas and non-Mendelian effects, but the environmental effects also have a big job. Then there's a term called epistasis, where more than two genes are maybe involved in making a trait appear a certain way. So realize that genes have all sorts of different jobs. In some cases, there's some genes for making a trait by making the pigment, or be intricately involved in making a pigment. These would be complex biochemical pathways. And other genes might be have a job of putting the pigments into the tissues, for instance. So we call this epistasis, where the interaction of two genes modifies the phenotype. So it's a little bit like continuous effect and pleiotropic and all those other things that we mentioned before, or up here, polygenic. So it's kind of like polygenic, but where more than one gene's involved. But this is a more straightforward example. This was discovered in 1918 by the geneticist Emerson, who crossed true breeding corn varieties with white kernels. And to his surprise, they were all purple, which kind of looks like incomplete dominance, right? And then he crossed and had another generation. All of a sudden, he sees a lot more whites, which doesn't make any sense to him, because they should have been a dominant recessive kind of thing. So the traits don't fit the Mendelian expectation, like you would have seen with the pea plants. because the Mendelian prediction would have been a 9-3-3-1 ratio. And in this case, he saw a 9 purple to 7 white ratio. So it didn't fit the Mendelian. And so what was the reasons for this modification? Well, again, there are 2 genes that contribute to kernel color. And again, what happens is it masks the appearance of Mendelian genetics. Here you have, instead of just, and just looking at 2 different genes, A and B, for instance, or B, you see that in the case of this picture here, we have 2 white corn being crossed, and then all of a sudden we see a purple. And why is that? Is that because of something about the pigments or whatever? Well, what we find out is that A is important for depositing the pigment and B is important for making the pigment. So it's both of these genes that are involved. So it's not a simple, look at A is dominant and big A is dominant and little a is recessive, like would have been expected. Does that make sense to everybody? It's multiple genes involved. And so here we show what the gametes would look like in our Punnett Square, and it's now got 16 boxes that represent these 2 different genes that are involved in these traits. So in the F generation, they're all purple. So we come up with all the different possible gametes where we have big A, big B, big A, little A, big B, little a, little b. And then we see the same possibilities on the left side for the eggs. It'd be pollen and so forth. We cross them together and we get these different combinations that results in this 9-to-7 ratio. Why? Well, look at what the genes are showing. So in this case, we got big A, big A, big B, big A, big A, big B, big B. That means that we're going to have the purple trait because the plant was able to make the pigment purple and deposit the pigments. Now look over here. We have big A, big little b, but it's white. Why is it white? Well, in this case, the purple pigment is a white, well, the white pigment's being made and is being deposited. What about little a, little b? In case the recessive genes show up, it's white again. What about big A, big B, little b? Well, it would normally, it's all purple again, just like in the F1 generation. So again, to see these colors, you need to have both of them present and in the dominant trait. So let's look at another example. Here we have big A, and well, I'm trying to find another one I like better. So here we have a big A and little b's. Let's see, do we have a big B anywhere? Here we have big B's and is able to be deposited. Oh, wait a minute, look at this one right here. 2 big B's. So what pigment was this corn plant making if it has 2 big B's? Purple, right? But it's not able to deposit because the A's are present. Does that make sense, everybody? So what's the take-home message? It took these 2 different sets of genes to mask what's happening with the Mendelian genetics that we normally think of, and we just look at 1 gene dominant and 1 gene recessive. That kind of makes sense a little bit? Oh, here's codominance. And this is one of those that gets kind of confusing and because it looks like incomplete dominance, doesn't it? Here we have 2 genes, maybe, I'm not sure which ones would have necessarily been, but here we have maybe a white gene and a black gene. And now we have a row. Well, what you'll see is that it has white fur and black spots. So we call this codominance when both traits are showing up. It's a little different than incomplete dominance, where one trait is is reduced in its effect, like the red flowers were not as brilliant red because of the white gene being present. But in the case of this roan horse, it kind of has a gray appearance, but it's actually due to white pigments and white pigment all being kind of interdispersed. So they're both being expressed. So it's a lot like incomplete dominance, but it's black dots and white are being expressed equally. And so we call that codominance. Does that make sense? And we see this also in regards to blood types. We have our A, B, and O blood types. The A and the B are both codominant. So that's why you can have A and B blood type. Or you can have an A blood type, and depending on your parents. So if you had an O negative, or say, let's not worry about the positive, but say you had an O positive father and an A mother, she's positive too, let's just keep it. You could have A blood. If she is homozygous A, meaning she has two A chromosomes, then you're guaranteed to be A if she's with an O dad. Because O is meaning you don't have that trait. Does that make sense, everybody? But if she's heterozygous, so she has a big A and O, and she hooks up with your dad who's O, then you could be O and or A. Does that make sense, everybody? Because that is also another codominance trait. And so again, O is means that you're lacking that phenotype. And then what are the phenotypes? There's sugar molecules and lipids on the surface of red blood cells. A adds a galactosamine, B adds galactose, and I, or the O, has neither. And so this is an example of the different types of phenotypes you could have, depending on your parents. So like if your mom was AB, and she hooked up with your dad who was O, and lacking him, you could be A or B, but you won't be AB. Does that make sense? Just following the Punnett Square. So let's say because your mom was AB, she would make gametes that were A or B, and her dad would only make I in that case, or O. And that means that you would only be able to be A or B. Now, if your mom was B, and she's BO, that doesn't sound right, then you could be, this depends on, so again, is this a combination? But again, it's another codominant trait. A and B are both expressed. And that again throws out the typical Mendelian genetics. The idea of chromosomes becoming part of inheritance was later formulated by Walter Sutton in 1902. So again, remember Mendelian genetics, back when Mendel was looking at that, he would just look at them as factors. He didn't know or realize that they were related to the chromosomes necessarily. So Walter Sutton in 1902 was able to start putting together the idea that the Mendelian factors were found on the chromosomes, and that these were independently sorted and helped explain a lot of what he was seeing. This was further established and figured out in 1910 by Thomas Hunt Morgan, who you can thank if you ever do fruit fly research or so forth. He was big into figuring out, looking at fruit fly traits, how chromosomes were involved. So Morgan tested these ideas in Mendelian fashion, looking at Drosophila. And again, they're easily among the most important organisms ever studied in biology when it comes to understanding genetics. So he would do all sorts of different crosses where he would compare females and males of different traits. Like here's a white-eyed male fly and a red-eyed female fly. Then he would just do a cross kind of like what you would see with Mendelian genetics, and then he would be able to make a prediction of what the, he would look at the chromosomes and make a prediction of what was the next generation. In this case, they're all red-eyed. And then he would do another cross in the second generation, see that we had that 3-to-1 ratio. So again, he was able to help kind of get and correlate how the chromosomes were involved in those traits. So anyway, he also, and so again, what he's looking at is sex-linked traits. That was one of the keys to figure out whether or not it was on a chromosome, because you know how there's X and Y chromosomes. So he was able to look at the Y chromosome to be able to establish that this trait must be on the Y chromosome or the X chromosome. So this helped provide evidence that chromosomes were critical for these factors, these Mendelian factors that we discussed. And so they were called sex-linked when they're found on the female chromosome, for instance, and not on the male chromosome. All right, so we've already talked about meiosis and chromosomes and there are things that sometimes happen when meiosis takes place where the chromosomes do not evenly divide. Remember when we talked about meiosis? We mentioned how if you divide cells that you should end up with 4 haploid cells if it's sperm. And for the case of the egg, you'll have 4 haploid cells again, but only 1 egg with half the number of chromosomes, and the other 3 are they just kind of thrown away or utilized as part of the support cells. Well, they don't always separate perfectly during meiosis. And in some cases, you can end up with extra chromosomes. And when you end up with extra chromosomes and that egg gets fertilized or that sperm has an extra chromosome, you can end up with some genetic disorders like Down syndrome. And that's due to the non-disjunction of the chromosomes during meiosis 1 or 2. And so this abnormal number of chromosomes are called aneuploidy. Remember, you're supposed to have 23 pairs of chromosomes. Well, some people, they could have an extra chromosome or 1 less chromosome. Humans with 1 less autosome are called monosomics, but they typically do not survive development. Humans with 1 extra chromosome are typically called trisomics. The vast majority do not survive, but some trisomies work in some chromosomes, but typically have developmental defects. The one that is most successful is the trisomy of chromosome 21. So here's a person's karyotype who has Down syndrome, who has an extra chromosome on 21. And so she has Down syndrome. And again, this is doing this based on meiosis and not having a proper dissociation of one of the chromosomes. The eggs divide better during for a younger mother. So typically 1 in 1,500 mothers under 30 may have a child with Down syndrome, but it ends up being 1 in 16 for mothers over 45. And there's probably some, and this is, I think, also can be related to really older fathers too, to some degree. Then there's also non-disjunction that takes place in sex chromosomes as well. In some cases, this can lead to different phenotypes, and sterility. So there can be situations where you can have gametes with two Ys and then XXY zygotes. So so you can have individuals with men with an extra Y chromosome. I believe there's some evidence that men with extra Y chromosomes tend to have more testosterone and more likely to be found in the prison system than the typical XY man. To a certain percentage, I don't know what it is, but something like that. There are some females that can have triple X chromosomes or 1X and no chromosome. There could be men with an extra X chromosome and they tend to have a little bit more of a feminine trait. That's known as Klinefelter syndrome. Or a man without an X chromosome. So this is the non-disjunction that takes place with Y chromosomes and X chromosomes. But here's an example with the non-disjunction taking place in the egg. So those are some of the kind of classical examples of non-disjunction, how that affects certain traits. Mutations are also a big part of genetics and a big part of evolution, as we talked about, where a gene can be altered, where the different nucleotides get changed out, and that might bring in a different amino acid. Remember how I talked about there's 3 different nucleotides involved, and then they ultimately can come together to make a certain amino acid come in. Well, those are examples of mutations. A lot of times mutations are harmful and typically cause genetic disorders. One example was the cystic fibrosis I talked about, where there could be a mutation that takes place that makes the chloride transport channel not work properly and make the lungs and fluids not work too well either, and it gets clogged up. We can also see things like hemophilia take place. And so you can figure out genetic disorders by looking at the pedigrees and family trees of individuals, and then try to piece together who may have been the carrier of a genetic disorder, which typically are recessive traits. So a female is listed as being a circle, men are listed as a square in this particular pedigree that they put together. And they find that this male, for instance, is going to be affected by this genetic disorder because both of his parents were carriers of that genetic disorder, even though neither one of them showed the traits. So again, it's going to follow kind of a Punnett Square, and this female here is going to not show the trait, and then there's going to be 2 hybrids who are not affected, but are carriers. Does that make sense to everyone? This was a bigger issue in royalty, for instance, who tended to be very selective of who would hook up with their family, right? So they would tend to have some inbreeding going on, or royal to keep the money together and not. So they would do a lot of, I hate to call it inbreeding, but that's what it was, and increase the likelihood of seeing sex-linked traits like hemophilia. Hemophilia is the inability to really be able to clot your blood properly and cause you to bleed to death. So this was typical, or could happen, or was shown, like for instance, in England, where the Queen Victoria had many children that had this defective allele. And again, it was a sex-linked trait, so it was more likely the boys would have hemophilia because the trait would show up on the X chromosome. And then there was, on the Y chromosome, there wasn't any corresponding gene to the X chromosome to fend off that trait. So if it was up on the X chromosome, there wasn't a corresponding chromosome on the Y chromosome. So that recessive trait could show up. Does that make sense, everyone? Because if it was two X chromosomes, you could have the recessive trait for hemophilia, but you could also have a healthy gene to prevent hemophilia in a female. So a heterozygous female would just be a carrier and not show hemophilia. And if you had an X chromosome with the recessive trait and a Y chromosome, then you could see the trait in the male. So men are more likely to get often sex-linked traits if it's showing up on the X chromosome, obviously. And again, because of the inbreeding that showed up, hemophilia showed up more and more. So they did a pedigree on 10 of Queen Victoria's male descendants, finding out who had hemophilia. When Queen Victoria herself was a carrier, hooked up with Prince Albert, Alice was an example of someone who was a carrier, and these individuals weren't. And then Irene was a carrier, and then I guess she ended up having children. Looks like she had two different fathers, but or husbands. They ended up with children, males that had hemophilia, and one didn't. So the hemophilia trait was being interbred because, again, royalty was kind of breeding with royalty that had that trait, and so it was more likely to show up. So that's how in a recessive trait can show up more if you end up with other individuals that are carriers and the interbreeding of those individuals, because they're all part of that same status. They're not going down to another village and picking up a woman or a man, you know what I'm saying? They're going to stick with another prince. Here's also colorblindness is also a sex-linked trait, where more men are colorblind than women. And it has to do with the fact that that gene is showing up on the X chromosome and the Y chromosome has nothing to counteract it. So colorblindness is actually a recessive trait, and plus is a normal vision. So men who have the X chromosome for colorblindness have no corresponding gene on the Y chromosome, and so they are colorblind. So they need a mother that gives them the normal gene, and then they're going to have normal vision. While females are only colorblind if they get two X chromosomes with the defective gene. Maybe there's a benefit for colorblindness in some cases. I should be careful what we're using it, but the normal trait would be considered, and the recessive trait is colorblindness. Again, that's going to be due to some type of mutation. Sickle cell anemia is a recessive trait where this is a blood cell that typically looks like a lifesaver. It's full of hemoglobin. A normal one can move easily through capillaries. But those with sickle cell anemia, the blood cell does not have a proper shape. It actually has a sickle shape and gets caught up and clogged up in blood vessels, increasing the likelihood of a shortened lifespan with lots of intermittent illnesses. But there is also a heterozygous trait, which is actually a carrier, which is somewhere in the middle. Maybe it still has a normal looking blood cell, but it actually helps them to prevent them from having malaria. So there's actually a benefit to the heterozygous trait being a disease carrier. So again, sickle cell anemia is is in this case an autosomal recessive trait. Remember the other ones that we talked about, like with color blindness and hemophilia, was a sex-linked. Autosomal means those normal chromosomes that are floating around in your cells, your adult cells, that are involved with sex traits directly. Sickle cell anemia is defective. It's a protein that changes the shape of the hemoglobin. Hemoglobin is the protein that transports oxygen. Interestingly enough, those with sickle cell trait are more likely to survive malaria. So there may be a benefit to having this be a disease carrier in countries or continents where malaria is higher in amounts. Malaria is a disease that's spread by mosquitoes that spreads a protozoan into your blood. So people from Africa are more likely to have sickle cell traits due to having selective pressure from malaria. There's other types of recessive traits that are also diseases, like Tay Sachs disease is an autosomal recessive trait that causes the enzyme hexosaminidase A to be defective and prevent those individuals from being able to break down lipids. Typically, children around the age of 5 die from Tay Sachs disease. So it's heavily selected out of the environment, out of our population, because those people don't go on to get to breed, right? So those type of recessive diseases tend to be selected out of our population. Darwinianly, natural selection, select them out. However, there are some populations that do have higher amounts of it than others. However, Huntington's disease shows up more often in populations, even though it's a dominant trait, because it happens after people reproduce. So some diseases will still show up more often, like Huntington's disease, because in the population, because people are able to reproduce, happens after they've had that reproductive stage. So some disease genes like Tay Sachs is selected away from the population through evolution, natural selection, because those individuals can't reproduce, while some can still be present because they stay in the population because reproduction is so late in onset. Some traits are autosomal, but they're sex influenced. That also is interesting. So, like, as we all know, men tend to be more bald on average than women because it is a sex influence trait, not a sex linked. So this is a gene that does show up on autosomal chromosomes, not the sex chromosomes. But because there's testosterone made by men, it affects the presence of that expression differently. So men that are recessive for baldness tend to have normal hair irrespective of their testosterone levels. While other men, if they receive the baldness chromosomes from the mother and father, will start to see their hair thin, whether they're heterozygous or they'll be bald. Does that make sense? So this individual is going to be bald and this one's going to be bald. While females, even if they have the genes, they're only going to be bald if they're homozygous for the baldness gene, not heterozygous, because they're not showing as much testosterone. So this is a sex influence trait. One area that's going to become more and more important that you may want to consider for a career someday is genetic counseling. The more and more we're learning about genes, the more we're going to have to be able to counsel people on what that means, educate them. So genetic counseling is going to continue to become a more important thing. Knowing your genes before you reproduce with somebody might be an important thing too, to make sure you know if you're with somebody, is there a certain odds or percentage chance that your children will have cystic fibrosis? Those are things that may be worthwhile to let you know before you decide to have children. So genetic counseling can be important for those kind of things as well. You know, is your pregnancy going to be a more high risk? Is that more likely, you know, after 40 or 35 or 40 or 45, pregnancies become more high risk? You're more likely to have trisomies and things like that. Those are all things that genetic counseling and therapy should be discussed, and it's going to become more important as time goes on. We can get an idea of the health of our baby by doing amniocentesis, where they'll take a little bit of amniotic fluid with a needle, and then they can look at the fetal cells and get an idea of whether or not the fetus is growing normally or has any of these genetic issues. So this is typically done particularly for later term pregnancies. I don't know if it happens for younger pregnancies or not. It's been a while since I've been involved with a pregnant woman. But I remember, I think at least once she did something like this. Ultrasound is also important to look at whether or not the fetus is developing normally. So we have all sorts of great tools now to help in watching all the steps of pregnancy and then having an idea of what's happening with the baby before it comes out. There's other methods too, like chronic villus sampling. I don't know that much about it. And then there's ways of looking at the karyotypes and enzyme activities and genetic markers. So there's all sorts of ways of screening pregnancy in babies and trying to see what their genetic issues may be. And there's other types of DNA screening that's becoming more interesting or important. I don't know, again, a whole lot about it, but you can look at single nucleotide polymorphisms, which are ways of screening for mutations and trying to figure out genetic disorders and things like that as well. So in general, that kind of wraps up what you need to understand for Mendelian genetics and some other aspects of genetics in regards to zoology. I appreciate you paying attention. Realize that we're gonna probably have to start thinking about an exam. So I will try to get you a study guide and a plan for that. Anyway, thanks for listening, and you all take care.